Finished thinking. Microscopic techniques are methods used to observe objects and structures that are too small to be seen with the naked eye. These techniques are essential in fields like biology, materials science, and medicine. Here's a quick breakdown of the main types. 1. Light microscopy. Uses visible light and lenses to magnify samples. Types. Bright field microscopy. Phase contrast microscopy. Fluorescence microscopy. Confocal microscopy. 2. Electron microscopy. Uses beams of electrons for higher resolution. Types. Transmission electron microscopy TEM gives detailed internal structure. Scanning electron microscopy SEM shows surface structure in 3D. 3. Scanning probe microscopy uses a physical probe to scan the surface of a specimen. Types. Atomic force microscopy AFM. Scanning tunneling microscopy STM. 4. Other advanced techniques. Cryo-electron microscopy, used for imaging biological molecules in near-native states. Super-resolution microscopy, breaks the diffraction limit of light e.g., storm, palm. Do you want a deeper dive into one of these or how they're used in a specific field? You said. For CSIR Net-oriented theory lectures and previous year questions answers with year in bracket high-quality lecture ChatGPT said. Memory updated. Here's a detailed CSIR Net Life Sciences-oriented theory lecture on microscopic techniques, including previous year questions with answers with years mentioned. Microscopic techniques, CSIR Net Life Sciences theory lecture. Microscopy is a central tool in life sciences, enabling the visualization of cells, tissues, and macromolecules. The choice of microscopy depends on the nature of the sample, required resolution, contrast, and whether live imaging is needed. 1. Light microscopy. Principle, uses visible light passed through or reflected from a specimen and magnified through optical lenses. Types. Bright field microscopy, the simplest form where light passes directly through the sample. It's suitable for stained specimens. Phase contrast microscopy, enhances contrast in transparent specimens by exploiting differences in refractive index. Ideal for live cells. Differential interference contrast stick microscopy, uses polarized light for 3D-like images of unstained cells. Fluorescence microscopy, uses fluorophores that emit light upon excitation, widely used in cell biology to detect specific proteins or organelles. Confocal laser scanning microscopy, uses laser beams and pinholes to eliminate out-of-focus light, enabling optical sectioning and 3D reconstruction. Applications, localization of proteins, visualization of organelles, and live cell imaging. 2. Electron microscopy EM. Principle, uses a beam of electrons instead of light, hence resolution is much higher due to the shorter wavelength of electrons. Types. Transmission electron microscopy TEM. Electrons pass through ultra-thin sections of the specimen. Provides detailed images of internal structures at the nanometer scale. Scanning electron microscopy SEM. Electrons scan the surface of a specimen. Produces detailed 3D images of surfaces. Limitations, requires vacuum conditions and fixed dead samples, preparation is time-consuming. Applications, high-resolution imaging of viruses, cellular organelles, and tissues. 3. Scanning probe microscopy SPM principle, uses a physical probe to scan the specimen surface. Types. Atomic force microscopy AFM. A cantilever with a sharp tip physically interacts with the sample. Measures deflections to construct topographical maps. Scanning tunneling microscopy STM. Measures tunneling current between probe and sample. Only works with conductive surfaces. Applications, imaging biological macromolecules like DNA, membrane proteins, and cell surfaces at atomic resolution. 4. Super-resolution microscopy. Need, conventional light microscopy is limited by the diffraction limit, approximately 200 nanometers. Super-resolution techniques surpass this limit. Techniques. Storm, stochastic optical reconstruction microscopy. POM, photo-activated localization microscopy. Principle, involves sequential activation and precise localization of fluorophores. Applications, imaging molecular architecture of cells with nanoscale precision. 5. Cryo-electron microscopy, cryo-EM. Principle, samples are rapidly frozen to preserve native structure and image at cryogenic temperatures in TEM. Revolutionized structural biology, particularly in understanding the structures of large complexes like ribosomes, viruses, and membrane proteins. CSIR Net-oriented key points. Know the resolution limits of each technique. Light, approximately 200 nanometers, TEM, approximately 0.2 nanometers, SEM, approximately 10 nanometers, AFM, approximately 1 nanometer. Understand the advantages, disadvantages, e.g., live imaging possible with phase contrast and fluorescence, not with EM. Identify components of each microscope and their functions. Applications in molecular biology, cell biology, and biophysics are frequently asked. Previous year CSIR Net questions 1. CSIR Net Deck 2018. Which of the following microscopy techniques would you use to visualize live, unstained cells in culture? A. Bright field microscopy. B. Phase contrast microscopy. C. Scanning electron microscopy. D. Transmission electron microscopy. Answer. B. Phase contrast microscopy. 2. CSIR Net June 2016. A researcher wants to examine the surface structure of pollen grains. Which of the following techniques is most appropriate? A. Transmission electron microscopy. B. Bright field microscopy. C. Scanning electron microscopy. D. Fluorescence microscopy. Answer. C. Scanning electron microscopy. 3. CSIR Net Deck 2017. Which microscopy technique would allow optical sectioning and reduce out-of-focus light in thick specimens? A. Phase contrast. B. Bright field. C. Confocal microscopy. D. TEM. Answer. C. Confocal microscopy. 4. CSIR Net June 2019. What is the approximate resolution limit of light microscopy? A. 200 micrometers. B. 200 nanometers. C. 20 nanometers. D. 0.2 nanometers. Answer. B. 200 nanometers. Would you like the next lecture on cell fractionation and separation techniques or go deeper into confocal and super-resolution microscopy? You said. Visualization of cells and subcellular components by light microscopy. ChatGPT said. Here's a CSIR Net Life Sciences-oriented detailed theory lecture on the topic. Visualization of cells and subcellular components by light microscopy. Microscopic observation of cells and their internal structures is fundamental to understanding cellular function. Light microscopy remains the most commonly used tool for this purpose, particularly for visualizing live cells and dynamic processes. Principle of light microscopy. Light microscopy uses visible light wavelength approximately 400 to 700 nm to illuminate specimens and optical lenses to magnify the image. The resolving power is typically approximately 200 nanometers due to the diffraction limit of light. The image formation depends on light absorption, reflection, and refraction by the specimen. 2. Key components of a light microscope: light source, usually a halogen or LED lamp. Condenser, focuses light onto the specimen. Objective lens, gathers light from a specimen and magnifies it. Eyepiece, ocular lens, further magnifies the image. Stage, platform to place the slide. Coarse and fine focus knobs, adjust the distance between the specimen and the objective. 3. Type light microscopy for visualizing cells and organelles. 1. Bright field microscopy. Description, light passes directly through the specimen. Use, stain cells, commonly used in histology and microbiology. Limitation, poor contrast for unstained cells, limited resolution. 2. Phase contrast microscopy. Description, converts phase shifts in light passing through a transparent specimen into differences in light intensity. Use, visualizing live, unstained cells, ideal for observing organelles like nuclei, mitochondria, and vacuoles. Advantage, enhances contrast without staining. 3. Differential interference contrast stick microscopy. Description, uses polarized light to produce high contrast images with a 3D appearance. Use, detailed imaging of live cells and organelles. Advantage, improved resolution and depth perception. 4. Fluorescence microscopy. Principle, fluorophores absorb light at one wavelength and emit at a longer wavelength. Use, detection of specific cellular structures using fluorescent dyes or proteins, e.g., DAPI for DNA, GFP-tagged proteins. Applications. Localizing proteins within cells. Tracking dynamic processes, e.g., mitosis. Multicolor imaging of multiple organelles simultaneously. Multicolor imaging of multiple organelles simultaneously. 5. Confocal laser scanning microscopy. Description, uses lasers and pinholes to capture images from a single focal plane, eliminates out-of-focus light. Use, 3D reconstruction of cells and tissues. Advantage, high resolution and contrast, used for thick specimens or tissues. IV, staining techniques to visualize subcellular components: hematoxylin and eosin, H&E, general cell staining. DAPI or host, nuclear DNA. Mitotracker, mitochondria. Colloidin, actin filaments. Acridine orange, nucleic acids. Live dead stains, viability assays. Stains and fluorescent dyes allow specific targeting of organelles and biomolecules, enhancing both contrast and specificity under the microscope. V applications in cell biology. Visualization of nucleus, cytoskeleton, mitochondria, chloroplasts, lysosomes, endoplasmic reticulum. Studying cell division, apoptosis, membrane dynamics. Live cell imaging to monitor real-time processes. Pathological diagnostics and cytogenetics. Limitations of light microscopy. Limited resolution, approximately 200 nanometers, cannot resolve structures like ribosomes or macromolecular complexes. Photobleaching and phototoxicity in live cell fluorescence imaging. CSIR net-oriented key points. No types of microscopy and their advantages, limitations. Understand staining methods and specific dyes for organelles. Differentiate between contrast-enhancing methods, phase versus DIC. Recognize when to use confocal versus standard fluorescence microscopy. Previous year CSIR net questions. 1. CSIR net DEC 2018. Which of the following microscopes is best suited to observe live, unstained mammalian cells? A bright field microscope. B phase contrast microscope, CTEM. DSEM. Answer, B phase contrast microscope. 2. CSIR net June 2021. A scientist wants to study the subcellular localization of a specific protein in living cells using GFP. Which microscopy technique should be used? A bright field. B phase contrast, C fluorescence, DSEM. Answer, C fluorescence microscopy. 3. CSIR net DEC 2016. Which staining technique is commonly used to specifically label mitochondria in live cells? A DAPI. B acridine orange, C mitotracker, D DIMSA. Answer, C mitotracker. Would you like the next lecture on transmission and scanning electron microscopy or more solved MCQs on microscopy? You said, resolving powers of different microscopes, ChatGPT said. Here's a CSIR net life sciences oriented detailed lecture on the resolving powers of different microscopes, tailored for deep conceptual understanding and exam preparation. Resolving power of different microscopes. What is resolving power? Resolving power, also called resolution, is the ability of a microscope to distinguish two closely spaced objects as separate entities. It determines the level of detail visible in the image. Formula for resolution: D equals 0.61 times lambda Na, where D equals minimum resolvable distance. Lambda equals wavelength of light or electrons. Na equals numerical aperture of the objective lens. Smaller d value equals higher resolution. 1. Light microscopes. A bright field microscopy resolution, approximately 200 nanometers nm. Limitation, cannot resolve structures smaller than 200 nanometers such as ribosomes or viruses. Reason, limited by the wavelength of visible light, approximately 400 to 700 nm. B phase contrast in DIC microscopy resolution, also approximately 200 nanometers. Purpose, enhance contrast, not resolution. Application, best for viewing live, unstained specimens. C fluorescence microscopy resolution, approximately 200 nanometers, diffraction limited. Limitation, fluorophore spread and light diffraction limit precise resolution. D confocal microscopy. Resolution, approximately 180 to 200 nm in XY plane, approximately 500 nanometers in Z plane. Advantage, optical sectioning and improved image clarity using pinhole aperture and lasers. 2. Super resolution microscopy, diffraction unlimited light microscopy, breaks the 200 nanometers diffraction barrier using advanced techniques. ASTED stimulated emission depletion microscopy resolution, approximately 20 to 50 nm. Technique, uses depletion lasers to sharpen the illumination point. B storm and palm resolution, approximately 10 to 20 nm. Technique, sequential activation and localization of single molecules. Application, imaging cytoskeletal filaments, membrane proteins, and macromolecular complexes at near molecular resolution. 3. Electron microscopy, EM. Electrons have much shorter wavelengths than light, offering much higher resolution. A transmission electron microscopy, TEM resolution, approximately 0.1-0.2 nanometers. Application, internal structures of cells, organelles, ribosomes, macromolecules. B scanning electron microscopy, SEM resolution, approximately 1 to 10 nanometers. Application, surface topology and 3D imaging. 4. Scanning probe microscopy, atomic force microscopy, AFM resolution, approximately 1 nanometer lateral, approximately 0.1 nanometers vertical. Application, DNA, proteins, membrane structures at atomic resolution. B scanning tunneling microscopy, STM resolution, sub-nanometer, down to 0.01 nanometers. Limitation, requires conductive surfaces. Summary of resolving powers, bright field, phase contrast, DIC, approximately 200 nanometers. Fluorescence, approximately 200 nanometers. Confocal, approximately 180 to 200 nm. STED, storm, PALM, super resolution, approximately 10 to 50 nm. SEM, approximately 1 to 10 nm. TEM, approximately 0.1 minus 0.2 nanometers. AFM, approximately 1 nanometer lateral, approximately 0.1 nanometers vertical. STM, approximately 0.01 nanometers. CSIR net-oriented key points. Resolution limit of light microscopy, 200 nanometers frequently asked. Super resolution and EM techniques are vital for visualizing viruses, ribosomes, and proteins. No approximate resolution values in their order of magnitude. Electron microscopes provide resolution due to short electron wavelengths, not due to lens quality. Previous year CSIR net questions 1. CSIR net DEC 2017. Which of the following microscopy techniques offers the highest resolution? A bright field. B phase contrast, C transmission electron microscopy, D fluorescence microscopy. Answer, C transmission electron microscopy. 2. CSIR net June 2020. The resolution limit of a typical light microscope is approximately A 0.02 nanometers. B 2 nanometers, C 200 nanometers, D 2 micrometers. Answer, C 200 nanometers. 3. CSIR net DEC 2016. A scientist needs to visualize the detailed surface topology of a pollen grain. Which technique is appropriate considering its resolution? A bright field microscopy. B confocal microscopy, CSEM. D TEM. Answer, CSEM. Would you like the next lecture on fluorescence-based imaging techniques or a combined comparison of all microscopy techniques based on resolution, contrast, and application for quick revision? You said. Microscopy of living cells, ChatGPT said. Here's a CSIR Net Life Sciences oriented detailed lecture on microscopy of living cells, with full theory, key points, and previous year questions with answers. Microscopy of living cells. Understanding the structure, dynamics, and functions of living cells requires imaging techniques that preserve cell viability while offering sufficient resolution and contrast. Specialized forms of light microscopy have been developed to meet these demands. IY microscopy of living cells. To observe real-time biological processes, e.g., mitosis, cell migration, endocytosis. To study cellular responses to stimuli or drugs. To visualize dynamic interactions among proteins and organelles. To avoid fixation artifacts that occur in dead cells. 2. Microscopy techniques suitable for live cell imaging. 1. Phase contrast microscopy. Principle, converts differences in refractive index which affect phase of light into contrast. Best for unstained, transparent cells. Advantages. Visualizes organelles, nucleus, vacuoles, granules. Non-invasive, no dyes needed. 2. Differential interference contrast stick microscopy principle, uses polarized light and optical prisms to generate high contrast, 3D-like images. Advantages. Sharper contrast and phase contrast. Excellent for observing live cell edges and membranes. 3. Fluorescence microscopy, live cell fluorescence imaging, fluorophores used, GFP, RFP, CFP, etc. Application. Track specific proteins or organelles using fluorescent tags. Monitor dynamic events, e.g., protein trafficking, calcium flux, mitosis. Live cell compatible dyes, mitotracker mitochondria, calcium AM, viability, host nucleus. 4. Confocal laser scanning microscopy. Principle, uses laser light in a pinhole to collect focused optical sections. Advantages. Improved resolution and clarity. Enables time-lapse imaging in 3D Z-stacks. Ideal for thick specimens and tissues. Limitation, laser intensity may cause phototoxicity. 5. Spinning disc confocal microscopy. Faster and less phototoxic than traditional confocal, suitable for rapid imaging of moving structures in live cells. 6. Total internal reflection fluorescence TIRF microscopy application, imaging near the plasma membrane, e.g., exocytosis, receptor clustering. Principle, illuminates a thin section, approximately 100 to 200 nm, near the cover slip, reducing background. 7. Light sheet fluorescence microscopy, LSFM principle, uses a sheet of light to illuminate only a thin plane in a sample. Advantage, low photo damage, suitable for long-term live imaging in embryos or organoids. 3. Special considerations in live cell imaging temperature control, cells require 37 degrees Celsius for physiological behavior. CO buffering, especially in mammalian cells. Media, phenol red-free, low fluorescence background. Minimize photobleaching, use lower light intensity and anti-fade agents. Time-lapse microscopy, to observe processes like mitosis, cell division, migration, apoptosis, etc. IV, limitations lower resolution than M. Risk of phototoxicity and photobleaching. Need for special imaging chambers and environmental control. Fluorescent protein overexpression may alter natural cell function. V applications of live cell microscopy visualizing. Organelle movement, e.g., mitochondrial dynamics. Cell division stages. Intracellular transport, e.g., endosomes, vesicles. Calcium signaling using Fura2 or Fluo4. Membrane ruffling, lamellipodia, and filopodia. CSIR net-oriented key points. Phase contrast and DIC are preferred for unstained living cells. GFP tagging allows real-time visualization of proteins. Confocal interferorescence-based methods suited for live cell imaging. Phototoxicity and temperature, CO control are important for accurate observations. Live cell imaging equals dynamic, physiological processes, not static morphology. Previous year CSIR-NET Questions 1. CSIR-NET Deck 2018. A researcher wants to study the movement of mitochondria in live fibroblast cells. The best technique to use would be a bright field microscopy. B phase contrast microscopy, C fluorescence microscopy using NeoTracker, D transmission electron microscopy. Answer: C fluorescence microscopy using NeoTracker. 2. CSIR-NET June 2021. Which of the following techniques allows observation of real-time changes in protein localization in a living cell? A. Immunofluorescence after fixation. B. Electron microscopy of section tissue. CGFP tagged protein imaging using confocal microscopy. D. Histochemical staining of fixed samples. Answer: CGFP tagged protein imaging using confocal microscopy. B. Electron microscopy of section tissue. CGFP tagged protein imaging using confocal microscopy. D. Histochemical staining of fixed samples. Answer: CGFP tagged protein imaging using confocal microscopy. 3. CSIR-NET Deck 2016. Which microscopy technique allows selective imaging of fluorophores close to the plasma membrane in living cells? A. Phase contrast microscopy. B. Turf microscopy. C. Dick microscopy. D. TEM. Answer: B. Turf microscopy. Would you like the next lecture on fluorescence-based imaging techniques, or transmission electron microscopy? TEM. You said. Scanning and transmission microscope. ChatGPT said. Here's a CSIR-NET Life Sciences-oriented detailed lecture on scanning electron microscopy (SEM) and transmission electron microscopy (TEM), covering theory, applications, advantages, limitations, and previous year CSIR-NET questions with answers. Transmission electron microscopy (TEM) and scanning electron microscopy (SEM). Electron microscopy uses a beam of accelerated electrons instead of light to obtain ultra-high-resolution images. The very short wavelength of electrons, approximately 0.005 nanometers at 100 keV, allows sub-nanometer resolution, making them the tool of choice for visualizing ultrastructural details of cells and macromolecules. I transmission electron microscopy (TEM) principle. A beam of electrons is transmitted through an ultra-thin specimen. Electrons interact with the sample. Differences in density cause varying levels of electron scattering. The resulting image shows internal structures in high detail. Key components. Electron source, usually a tungsten filament or field emission gun. Electromagnetic lenses. Vacuum chamber. Specimen holder. Fluorescent screen or CCD camera. Resolution approximately 0.1-0.2 nanometers. Specimen preparation. Samples must be fixed, dehydrated, and embedded in resin. Ultra-thin sections 50-100 nanometers are made using an ultramicrodome. Staining with heavy metals, e.g., uranyl acetate, lead citrate, enhances contrast. Applications. Visualizing organelles, mitochondria, Golgi, ribosomes. Viewing macromolecular complexes. Virus morphology. DNA protein structures. Ultrastructural pathology. Advantages: very high resolution. Excellent for studying internal structures. Limitations. Requires thin, fixed, and dehydrated specimens. No live cell imaging. Expensive and time-consuming. 2. Scanning electron microscopy (SEM) principle. A focus beam of electrons scans the surface of a specimen. Secondary electrons emitted from a surface are detected to create a 3D-like image. Key components: electron gun. Scanning coils. Detectors for secondary and backscattered electrons. Vacuum chamber. Resolution approximately 1 to 10 nanometers. Specimen preparation. Fixed and dried commonly by critical point drying. Coated with a thin layer of conductive material, e.g., gold, palladium, or platinum. Applications. Surface topology of cells and tissues. Morphology of microorganisms, pollen, and insects. Industrial materials and nanoparticles. Advantages. 3D surface visualization. Greater depth of field than TEM. Easier sample preparation than TEM. Limitations. Surface only, cannot see internal structures. No live imaging. Requires conductive coating. 3. Comparison, TEM versus SEM. Feature: TEM SEM. Image type 2D, internal structures, 3D light, surface topology, resolution approximately 0.1-0.2 nanometers, approximately 1 to 10 nm, sample thin sections, surface contrast, internal ultrastructure, surface detail, use cell organelles, macromolecule cell surfaces, external morphology. CSIR-NET oriented key points. TEM equals internal structures, SEM equals surface details. TEM has higher resolution than SEM. Heavy metal staining is used in TEM. SEM needs metal coating to allow electron emission. Both require vacuum, no live imaging possible. No resolution ranges and appropriate applications for each. Previous year CSIR-NET Questions 1. CSIR-NET Deck 2018. To visualize detailed surface morphology of a nematode worm, which microscopy technique is most appropriate? A. Light microscopy. B. Transmission electron microscopy. C. Scanning electron microscopy. D. Fluorescence microscopy. Answer: C. Scanning electron microscopy. 2. CSIR-NET June 2017. Which of the following techniques would allow visualization of ribosomes in a liver cell? A. Bright field microscopy. B. Scanning electron microscopy. C. Transmission electron microscopy. D. Phase contrast microscopy. Answer: C. Transmission electron microscopy. 3. CSIR-NET Deck 2020. In electron microscopy, heavy metal staining is required because it increases brightness. B. It enhances contrast by scattering electrons. C. It stains live cells. D. It prevents beam damage. Answer: B. It enhances contrast by scattering electrons. Would you like the next lecture on fluorescence microscopy techniques, GFP, FRET, FRAP, or on sample preparation techniques for electron microscopy? You said. Different fixation and staining techniques for M. ChatGPT said. Here's a high-quality, CSIR-NET Life Sciences-oriented lecture on fixation and staining techniques for electron microscopy (EM), covering detailed theory, key applications, and previous year questions with answers in year mentioned. Fixation and staining techniques for electron microscopy (EM). Electron microscopy, both TEM and SEM, requires specialized sample preparation techniques, including fixation and staining, to preserve structural details and provide sufficient contrast. Unlike light microscopy, EM does not use colored dyes, but instead relies on electron-dense materials, heavy metals, to enhance contrast by scattering electrons. Eye fixation in electron microscopy. Fixation stabilizes biological structures by cross-linking proteins and lipids, preventing degradation or distortion during processing. 1. Primary fixatives glutaraldehyde. Fixes proteins by cross-linking amino groups. Provides excellent structural preservation. Used at 2.5-4% in phosphate or capitolate buffer. Formaldehyde, paraformaldehyde. Less cross-linking than glutaraldehyde, often used in combination. Mixture Karnofsky's fixative 2% paraformaldehyde plus 2.5% glutaraldehyde, for optimal preservation. 2. Post-fixation osmium tetroxide, OSO. Fixes membrane lipids and enhances electron density. Used after aldehyde fixation. Also acts as a stain by reacting with unsaturated lipids. 3. Fixation for SEM. Similar aldehyde fixation, glutaraldehyde followed by dehydration. No ultra-thin sectioning needed. Osmium tetroxide helps preserve and stain membrane surfaces. 2. Dehydration and embedding, mainly for temp dehydration, series of graded ethanol or acetone 30% to 100%. Embedding. Resin embedding, e.g., EPON, spurs resin, makes the sample rigid. Enables ultra-thin sectioning 50-100 nanometers using an ultramicrodome. 3. Staining techniques in EM. Staining in EM uses electron-dense heavy metals that scatter electrons to create contrast. 1. Staining for TEM, internal structures, uranyl acetate. Binds to nucleic acids and membranes. Enhances contrast in nucleus, ribosomes, and organelles. Lead citrate. Stains membranes and proteins. Improves overall contrast. Usually done post-sectioning on grid-mounted ultra-thin sections. 2. Negative staining TEM of isolated particles used for viruses, organelles, ribosomes. Phosphotungstic acid, uranyl acetate, or ammonium molybdate create contrast around the specimen. Background is dark, while the specimen appears light. 3. Staining for SEM, surface structures, no internal staining is needed. Surface staining with osmium tetroxide improves image quality. Metal coating gold, palladium, platinum using sputter coating makes the sample conductive. IV. Cryofixation and freeze substitution. Used for high-resolution preservation of biological structures in near-native states. Cryofixation, rapid freezing using liquid nitrogen or propane. Freeze substitution, replaces water with organic solvents at low temperatures. Preserves ultrastructure and avoids chemical fixative artifacts. DCSIR net-oriented key points glutaraldehyde, fixes proteins, used first. Osmium tetroxide, fixes lipids, used second, also a stain. Uranyl acetate and lead citrate, TEM staining agents. Gold coating, used in SEM for conductivity. Negative staining, TEM for viruses, ribosomes. Cryofixation, preserves native ultrastructure without chemical fixation. Previous year CSIR net questions 1. CSIR net Deck 2017. Which of the following fixatives is commonly used to preserve membranes in electron microscopy? A. Glutaraldehyde. B. Paraformaldehyde. C. Osmium tetroxide. D. Acetic acid. Answer: C. Osmium tetroxide. 2. CSIR net June 2019. Which stain is used in transmission electron microscopy to enhance contrast in nucleic acids and membranes? A. Safranin. B. Uranyl acetate. C. Crystal violet. D. Acecarmine. Answer: B. Uranyl acetate. 3. CSIR net Deck 2022. For scanning electron microscopy, the biological sample is usually coated with a silver nitrate. B. Osmium tetroxide. C. Gold or platinum. D. Bromophenol blue. Answer: C. Gold or platinum. Would you like the next lecture on fluorescence imaging techniques, GFP, FRET, FRAP, or light sheet and super resolution microscopy? You said. FreezeEtch. ChatGPT said. Here's a CSIR net life sciences oriented, high quality theory lecture on freeze etch, freeze fracture technique, including detailed concepts, applications, and previous year questions with answers in year mentioned, tailored for your exam preparation. Freeze etch, freeze fracture technique in electron microscopy. Introduction. The freeze etch or freeze fracture technique is a specialized sample preparation method used in transmission electron microscopy, TEM, for studying membrane architecture and the ultrastructure of biological membranes in their near native state. It avoids the chemical fixation and embedding artifacts seen in conventional EM methods. 2. Principle of freeze etch. The specimen, usually cells or tissues, is rapidly frozen to immobilize all structures instantly. Under a vacuum and at cryogenic temperatures, the frozen specimen is fractured, split using a microdome or blade. The fracture often occurs along the natural planes of weakness, especially through the hydrophobic core of lipid bilayers, revealing the internal faces of membranes. Etching. Some water is allowed to sublime, evaporate under vacuum, from the fractured surface, revealing more detail. The exposed surface is coated with platinum or carbon to form a replica. The biological material is then dissolved away, and the replica is examined under a TEM. 3. Key terminologies. P-face, protoplasmic face, the inner leaflet of the plasma membrane adjacent to the cytoplasm. E-face, exoplasmic face, the outer leaflet of the membrane facing the extracellular space. Intermembranous particles, indigo membrane proteins visualized as bumps or depressions on fractured membrane faces. IV. Advantages of freeze etch technique: avoids chemical artifacts from fixation and dehydration. Preserves native 3D membrane architecture. Excellent for studying membrane proteins, gap junctions, tight junctions, nuclear pores, and mitochondrial membranes. Allows quantitative and topographical analysis of membrane components. V. Limitations. Technically demanding and requires specialized freezing and fracturing equipment. Interpretation requires expertise. Only surface fracture views are possible, not internal volumes. Time-consuming and expensive. V. Applications. Studying membrane organization in plasma membranes, endomembranes, and organelles. Visualization of membrane-bound protein distribution. Investigating junctional complexes and transport vesicles. Used in neuroscience, cell biology, and virology. 7. CSIR net-oriented key points. Freeze etch is not a staining method but a physical fracturing and replication method. Fracture occurs between lipid leaflets, not across the membrane. Used mainly in TEM, not SEM. Sublimation of ice during etching improves surface visibility. Platinum carbon replica is image, not the biological specimen itself. Previous year CSIR net question, CSIR net deck 2021. Freeze fracture technique used in transmission electron microscopy is especially suitable for studying the cytoplasmic enzymes. B. Protein expression level. C. Membrane structure and protein distribution. D. Cytoskeletal organization using immunofluorescence. Answer. C. Membrane structure and protein distribution. Would you like the next lecture on super resolution microscopy techniques, STED, POM, STORM, or should we continue with fluorescent reporter techniques like GFP, FRAP, FRET? You said, freeze. Fracture methods for M, ChatGPT said. Here's a CSIR Net Life Sciences oriented, detailed theory lecture on freeze fracture methods for electron microscopy (EM), including principle, procedure, applications, and previous year questions with answers in year mentioned, written in a high quality, exam ready format. Freeze fracture methods for electron microscopy (MI) introduction. Freeze fracture electron microscopy is a specialized physical preparation technique used primarily with transmission electron microscopy (TEM) to visualize the internal architecture of biological membranes. This method involves rapidly freezing a sample and physically breaking it to reveal structural details of membranes that are otherwise difficult to resolve. 2. Principle of freeze fracture. The core principle involves splitting frozen cells along natural lines of weakness, typically the hydrophobic interior of lipid bilayers. The fracture plane passes between the two lipid leaflets of biological membranes, exposing the protoplasmic face P face, the inner monolayer adjacent to the cytoplasm. The exoplasmic face E face, the outer monolayer facing the extracellular space. This allows direct visualization of integral membrane proteins, seen as bumps or pits on the fractured surfaces. 3. Procedure of freeze fracture rapid freezing, cryofixation. The specimen is quickly frozen using liquid nitrogen or liquid propane to preserve ultrastructure instantly. Fracturing. In a vacuum at very low temperatures, the frozen specimen is cleaved with a microdome blade. Fracture occurs across membranes, splitting them along the lipid bilayer. Etching, optional freeze etching. Sublimation of surface ice under vacuum reveals more surface detail. Helps improve contrast by exposing structural features. Shadowing with heavy metals. A thin layer of platinum or platinum carbon is deposited obliquely to create a shadowed replica. A supporting layer of carbon is then applied vertically for strength. Replica cleaning and imaging. The biological material is digested away using acids or detergents. The metal replica is placed on EM grids and imaged using TEM. IV. Interpretation of freeze fracture images. P face, more intermembranous particles, integral membrane proteins. E face, fewer particles, smoother appearance. Junctions, gap junctions, tight junctions, and desmosomes appear as characteristic patterns. Nuclear pores and mitochondrial cristae can also be resolved. V. Applications. Studying the distribution and density of membrane proteins. Visualizing membrane specializations, e.g., gap junctions, tight junctions. Investigating membrane fusion, fission events. Exploring the architecture of organelle membranes, such as mitochondria and ER. Commonly used in neurobiology, virology, and cell biology. V. Advantages. High-resolution 3D visualization of membrane interiors. Minimal artifacts due to avoidance of chemical fixation. Provides both topographical and compositional information. 7. Limitations. Technically demanding, requires specialized cryo equipment. Image interpretation requires expert knowledge. Not suitable for routine or high-throughput applications. 8. CSIR net-oriented key points. Freeze fracture is a physical method, not chemical staining. Used in TEM, not SEM. Reveals intermembranous protein particles. Fracture occurs along the nonpolar core of lipid bilayers. P face shows more proteins than the E face. Involves cryofixation, fracturing, etching, and replica imaging. Previous year CSIR net questions 1. CSIR net deck 2021. Freeze fracture technique used in transmission electron microscopy is especially suitable for studying the cytoplasmic enzymes. B. Protein expression level. C. Membrane structure and protein distribution. D. Cytoskeletal organization using immunofluorescence. Answer. C. Membrane structure and protein distribution. 2. CSIR net June 2020. In freeze fracture M, intermembranous particles observed on fractured faces represent ribosomes. B. Cytosolic proteins. C. Membrane associated carbohydrates. D. Integral membrane proteins. Answer. D. Integral membrane proteins. Would you like the next lecture on super resolution microscopy techniques, STED, POM, STORM, or shall we move to fluorescence-based live cell imaging, GFP, FRAP, FRET? You said. Image processing methods in microscopy. ChatGPT said. Here's a high-quality CSIR Net Life Sciences oriented theory lecture on image processing methods in microscopy, covering key principles, techniques, applications, and previous year questions with answers in years, crafted for your exam level preparation. Image processing methods in microscopy. Introduction. Microscopy images, especially those obtained from advanced techniques like confocal, fluorescence, electron, and super-resolution microscopy, often require digital processing to enhance visibility, contrast, and quantitative interpretation. Image processing in microscopy involves the mathematical manipulation of digital images to extract or enhance biological information while minimizing noise and artifacts. 2. Objectives of image processing in microscopy. Enhancement, improve contrast, sharpness, or visibility of specific structures. Restoration, remove distortions or noise due to optics or detectors. Quantification, measure area, intensity, number of particles, fluorescence levels, etc. Segmentation, isolate and identify regions of interest, e.g., nuclei, cells, organelles. 3D reconstruction, generate 3D structures from 2D image stacks, e.g., confocal Z stacks. Colocalization analysis, determine spatial overlap of fluorescent markers. 3. Types of image processing methods of preprocessing techniques. Noise reduction, using filters like Gaussian blur or median filtering. Background subtraction, removes uneven illumination or autofluorescence. Normalization, standardizes brightness, contrast across image sets. B. Enhancement techniques. Contrast adjustment, expands dynamic range, e.g., histogram stretching. Sharpening filters, enhance edges, e.g., unsharp masking. Pseudo-coloring, applies artificial colors to grayscale images for better distinction. C. Segmentation techniques. Thresholding divides pixels into foreground and background based on intensity. Edge detection identifies object boundaries, e.g., Sobel or Canny edge detectors. Watershed algorithm segments overlapping or touching objects like nuclei. Deconvolution, a mathematical technique to reverse optical distortions caused by the point spread function (PSF) of the microscope. Widely used in confocal and wide-field fluorescence microscopy. Improves resolution and contrast, especially for 3D reconstructions. Quantitative analysis fluorescence intensity measurement. Object counting, e.g., number of nuclei, vesicles. Colocalization metrics, e.g., Pearson's correlation coefficient. Chymograph analysis for tracking movement across time. F3D image reconstruction. Using software, e.g., ImageJ3D, Amaris, Velocity to generate 3D models from Z-stacks. Essential for studying structures like neurons, tissues, or embryos. IV, software tools used in image processing. ImageJ3D, open source, highly customizable, supports plugins for segmentation, deconvolution, tracking. Cell Profiler, designed for high-throughput image analysis. Amaris, Velocity, commercial software for 3D and time-lapse imaging. MATLAB, used for algorithm development and automated analysis pipelines. SenseIce, LASX, Leica, manufacturer-specific image analysis platforms. V challenges in image processing. Overprocessing, can introduce artifacts or misrepresent biological reality. Photobleaching, phototoxicity, in time-lapse imaging. Bias, manual segmentation can be subjective. Data size, high resolution, 3D, and time-lapse images require significant storage and computing power. P. CSIR-net-oriented key points. Image deconvolution is used to improve clarity in fluorescence microscopy. Segmentation and thresholding are core to quantification. Pseudo-coloring does not represent actual wavelengths. Colocalization analysis is vital in dual-label fluorescence microscopy. 3D reconstruction is common in confocal Z-stack imaging. Image processing is a post-acquisition step, distinct from microscopy itself. 7. Previous year CSIR-net question, CSIR-net June 2019. Which of the following techniques is commonly used for improving the resolution of wide-field fluorescence microscopy images? A turf microscopy. B. Image deconvolution. C. Phase contrast microscopy. D. Dark field illumination. Answer, B. Image deconvolution. CSIR-net Deck 2020. In microscopy-based imaging, the process of separating objects from the background is referred to as a thresholding. B. Normalization. C. Pseudocoloring. D. Binning. Answer, a thresholding. Would you like the next lecture on super-resolution microscopy, STED, POM, STORM, or continue with fluorescence-based techniques, GFP, FRAP, FRET? You said. Electrophysiological methods, ChatGPT said. Here's a CSIR-net Life Sciences-oriented detailed theory lecture on electrophysiological methods, covering principles, types, key applications, and previous year questions with answers in years, written in high-quality academic style for your exam preparation. Electrophysiological methods. Introduction. Electrophysiology refers to the study of electrical properties of biological cells and tissues, particularly the generation and propagation of electrical signals. These methods allow real-time measurement of ion channel activity, membrane potentials, synaptic transmission, and neuronal excitability, making them essential in neurobiology, physiology, and pharmacology. 2. Fundamental concepts. Membrane potential arises from unequal distribution of ions Na, K, Cl, Ca squared across the plasma membrane. Electrical activity is primarily governed by ion channels, pumps, and transporters. Action potentials, synaptic potentials, and end-plate potentials are electrical events measured using these techniques. 3. Major electrophysiological techniques. 1. Intracellular recording. Measures the membrane potential inside a single cell using a microelectrode inserted into the cytoplasm. Can record resting potential, graded potentials, and action potentials. Applications. Neuron excitability studies. Cardiac electrophysiology. 2. Extracellular recording. Measures electrical activity from outside the cell or tissue. Records population activity or field potentials, e.g., EEG, ECG. Applications. Monitoring activity of large neuron populations. Recording action potentials from multiple neurons. 3. Patch clamp technique. A revolutionary method developed by Erwin Neher and Bert Sackman, Nobel Prize, 1991, to study ion channels in cells. Types. Cell-attached patch, electrode seals to membrane patch, monitors channel activity in native state. Whole cell patch, electrode breaks membrane, allows recording of entire cell's electrical activity. Inside-out patch, membrane patch is pulled away, cytoplasmic site exposed to solution. Outside-out patch, after whole cell configuration, membrane reseals, extracellular side faces bath. Applications. Ion channel biophysics. Drug screening for ion channel modulators. Synaptic transmission analysis. 4. Voltage clamp. Maintains a constant membrane potential while measuring ionic currents across the membrane. Helps study voltage-dependent gating of ion channels. Application. Determining activation-inactivation properties of specific ion channels. 5. Current clamp. Injects a known current into the cell and records the resulting change in membrane potential. Used to study how neurons generate action potentials in response to stimuli. 6. Multi-electrode arrays, MEAs. Records from multiple neurons or tissues simultaneously using an array of electrodes. Non-invasive, used in cultures or brain slices. Application. Network-level activity studies. Pharmacological testing. 7. Electroencephalography, EEG. Records electrical signals from the scalp to monitor brain activity. Reflects cortical activity and is widely used in cognitive neuroscience and clinical diagnostics. IV. Applications of electrophysiology. Study of neurotransmission and synaptic plasticity. Diagnosis of neurological and cardiac disorders. Understanding sensory transduction, vision, hearing. Drug development targeting ion channels. Investigating electrical coupling between cells. VCSIR-net-oriented key points. Patch clamp is the gold standard for ion channel studies. Voltage clamp measures ionic currents under fixed voltage. Current clamp measures voltage response under fixed current. EEG measures extracellular field potentials from brain. Field potentials represent the summed activity of multiple neurons. Whole cell recording allows access to intracellular contents for manipulation or measurement. P. Previous year CSIR Net Questions 1. CSIR Net Deck 2019. Which electrophysiological technique is most appropriate for studying the activity of a single ion channel in real time? A voltage clamp recording. B. Patch clamp in cell attached configuration. C. Field potential recording. D. Electroencephalography. Answer. B. Patch clamp in cell attached configuration. 2. CSIR Net June 2022. In a current clamp experiment, injection of depolarizing current into a neuron leads to a reduction in membrane potential. B. Hyperpolarization of the neuron. C. Initiation of action potentials. D. Decrease in sodium permeability. Answer. C. Initiation of action potentials. Would you like the next lecture on fluorescence-based imaging methods, PRAP, FRET, GFP, etc.?